welcome to Info Jungle, where the world's most remote and silent places are brought to life, where the slow unfolding of Earth's hidden wonders is an invitation to breathe a little deeper, to let the mind wander somewhere vast. Whether you are settling into bed right now, letting the day finally release its hold, sitting quietly with a warm drink as the hours grow late, or simply seeking company that demands nothing of you, we are glad you are here. Tonight's journey asks very little. It only asks that you stay a while, wherever you are as these words reach you. Somewhere out there, the night is already deep. Clocks have gone quiet. Streets have emptied. The particular stillness that arrives in late hours has settled. And far beyond wherever you are right now, farther south than most people ever think to look, there is a continent that has been holding this stillness, holding it for longer than the human mind can comfortably hold. Tonight, we are going to Antarctica, not just to the surface, not just to the white, wind-scoured expanse above, but deeper, into the ice, through it, down past layers of compressed time and buried atmosphere, into a world that exists in complete darkness, under immense pressure, sealed away from sunlight and open air for millions of years. A world where life, against every expectation, has continued. Quietly, patiently, magnificently. Before we descend, take a breath. Let the day go. Ask yourself this: what might be living in a place that has never seen light? What forms might life take after millions of years of isolation? What does it mean for something to survive? Not for a season, not for a lifetime, but for longer than human civilization repeated 10,000 times. These are the questions that Antarctica holds. They are not questions that demand quick answers. They are questions best approached slowly, the way the ice itself moves, with patience, with weight, with the understanding that some things reveal themselves only to those willing to wait. So settle in. The journey downward begins now. Antarctica is the kind of place that resists description, the way it resists human presence. Firmly, consistently, without apology. Most places on Earth can be reduced to a few reliable adjectives. Cold covers some of it. Remote covers a little more. But neither word captures what Antarctica actually is. It is something closer to a refusal of warmth, of familiarity, of the ordinary logic that governs most of the planet's surface. The continent covers nearly 14 million square kilometers. It is the fifth largest landmass on Earth, larger than Europe, larger than Australia, and yet it contains no cities, no permanent human settlements, no roads connecting one place to another, no bridges, no fields, no forest visible above the surface. It is governed not by any nation, but by an international treaty. Its most reliable inhabitants are researchers. They arrive in small groups for the southern summer months. Then they leave before conditions make staying impossible. The air itself communicates how seriously the continent guards its solitude. It is not merely cold. It is dry in a way that feels almost aggressive. A dryness that pulls moisture from exposed skin, from the insides of lungs, before the body has time to adjust. Antarctica is, technically speaking, a desert, not because of sand or heat, but because of precipitation. Some regions receive less snowfall in a year than the Sahara receives in rain. The wind compounds everything. It does not arrive in gusts the way wind does in familiar places. It arrives with what feels like intention. Steady, horizontal, scouring, moving not around things but through them. Equipment fails in Antarctic wind. Metal becomes brittle. Instruments freeze mid-reading. The body loses heat faster than every system designed to keep it warm. Sound behaves strangely here. In the open interior, where terrain stretches to the horizon without interruption, voices can carry for kilometers under the right conditions. The same conditions can swallow a sound entirely just meters away. There are no echoes to bounce off trees or buildings or hills. The ice absorbs and redirects everything. The silence of Antarctica is not the comfortable silence of a quiet room. It is a vast, physical, slightly disorienting absence. Researchers often describe it as one of the most unsettling experiences of their lives, and one of the most profound. Nearly 98% of Antarctica's surface is buried beneath ice. In some places, that ice reaches more than 4 kilometers deep. 4 kilometers of compressed, ancient snow, stacked layer upon layer over millions of years, pressing down with a weight that flattens mountain ranges beneath it, forcing the underlying rock deep into the Earth's crust. This ice is not static. It is not frozen the way a photograph is frozen. It moves, slowly, imperceptibly. Over the course of centuries, the ice flows outward toward the edges, carrying within it everything that has fallen on Antarctica. Since long before the earliest, humans walked elsewhere on the planet. And within that ice, something extraordinary is preserved. Tiny bubbles of atmosphere, trapped when ancient snow compressed into glacial ice. Hundreds of thousands of years ago, these bubbles hold samples of the air that existed at those moments. Scientists drill cores of ice from deep within the ice sheet. They read those bubbles the way a historian reads old letters, carefully, with an awareness that what is being examined is irreplaceable. These ice cores tell stories of ancient atmospheres, of carbon levels that rose and fell long before industrial civilization, of volcanic eruptions, of climate cycles stretching across ages. Ages that make recorded human history feel like a single short paragraph in a very long book. But beneath the ice lies something that science has only recently begun to understand. A hidden geography, a buried landscape that was once exposed to open air, shaped by rivers and glaciers and seas, home to forests and shallow oceans, home to organisms that no longer exist anywhere on Earth's surface. When Antarctica drifted southward and the global climate shifted, ice advanced and sealed all of that away. What remains below is not merely rock and sediment. It is a world that time and ice together decided to preserve. This is where the story of life beneath Antarctica stops being poetic and becomes scientific, because life did not simply vanish when the ice arrived. It adapted. It retreated into the cracks and the darkness and the cold, and found ways to continue. It has been continuing, quietly and without witnesses, ever since. If this slow descent into one of Earth's most hidden places is the quiet journey you needed tonight, consider subscribing to Info Jungle. There is always more to uncover, and we would love to have you along for the next exploration. Imagine descending through 4 kilometers of ice, not falling, descending slowly, the way a deep-sea submersible sinks through water, with the weight of everything above pressing down, the temperature around you holding at a steady, extraordinary cold. The light from the surface fades almost immediately. Within a few hundred meters, it is gone entirely. There is nothing to see. There is no sound except the very faint, very deep groan of ice. Ice under its own immense pressure. And then at the bottom of all of that ice, something appears. Something that should not be there. Water. Liquid water. Vast and dark and perfectly still. Sealed in complete isolation from the surface of the Earth. For somewhere between 15 and 25 million years. This is Lake Vostok, one of the most astonishing places on Earth. It lies beneath nearly 4 kilometers of the Antarctic ice sheet. In East Antarctica, it was discovered not by drilling, but by noticing something unusual. Radar surveys and gravity measurements showed the ice behaving differently. Above a certain region, the ice was floating, ever so slightly, on something fluid beneath it. When scientists understood what they were looking at, the implications took time to settle. This was not a small meltwater pocket. This was a lake the size of Lake Ontario, sealed beneath the ice, kept liquid by the immense pressure above, and by geothermal heat rising gently from the rock below. A lake that had been there undisturbed for longer than most modern mountain ranges have existed. The question that followed was quiet but profound. If liquid water has persisted in complete isolation for 25 million years, could life have persisted alongside it? The answer, when samples were finally retrieved, was more interesting than yes or no. It was yes, and we do not entirely understand what kind. The samples from Lake Vostok contained DNA sequences. Sequences that did not match any known organism in any scientific database. Some sequences were bacterial. Some appeared to represent more complex forms of life. Many remain only partially characterized years after discovery. Researchers debate the details with appropriate caution, checking and rechecking for possible contamination, subjecting the data to repeated analysis. But the pattern across multiple independent studies is consistent. There is biological activity in Lake Vostok. Life is there. It is doing something. And some of what it is doing does not fit neatly into any known category. Lake Vostok is one of more than 400 subglacial lakes. More than 400 lakes identified beneath the Antarctic ice sheet, scattered beneath kilometers of ice across an area the size of a continent, connected in some cases by networks of channels and slow-moving water, carrying dissolved minerals, chemical energy, and potentially living organisms from one sealed environment to another. These lakes range from small ponds to bodies that dwarf the Great Lakes of North America. Some are shallow and transient, periodically draining and refilling. Others are deep and ancient, sealed for timescales that challenge comprehension. What connects all of them is the absence of sunlight, not the dimness of deep water, not the weak filtered light of a deep forest floor. Complete, permanent, total absence of any light whatsoever. In such conditions, the biological strategies we are familiar with do not apply. Photosynthesis requires light. Without light, there is no photosynthesis. Without photosynthesis, there are no plants. Without plants, the food chains built on them collapse. Something else has to take their place. That something else is chemosynthesis, a process that existed on Earth long before photosynthesis evolved. Chemosynthetic organisms extract energy not from light, but from chemical reactions. They metabolize iron, sulfur, hydrogen, and methane, compounds that occur naturally in rock and water. They convert that chemical energy into biological matter, forming the base of food webs that operate entirely in darkness. This is how life functions near deep-sea hydrothermal vents, where scalding water rich in dissolved chemicals pours from the seafloor, supporting entire ecosystems of tube worms, crabs, fish, and microbes, organisms that have never once depended on sunlight. Something similar may be occurring in the subglacial lakes of Antarctica. Though the temperatures are far lower, the chemistry is different, and the energy available is orders of magnitude smaller. The implication, held quietly but seriously by researchers, is significant. Antarctica's subglacial lakes may contain ecosystems unlike anything described by science, not unlike anything imagined. Researchers are careful, measured, and appropriately skeptical, but unlike anything formally documented. Ecosystems operating on chemical energy alone, in complete darkness, under crushing pressure, cut off from the surface world for timescales so vast that evolution moved freely, unconstrained by competition from outside, undirected by the cycles of light and season that shape nearly every other form of life. The patience of it is what is hardest to hold in the mind. Not the cold, not the darkness, not even the pressure, but the time. 25 million years of quiet continuation. 25 million years of organisms metabolizing, reproducing, dying, being recycled, adapting by imperceptible degrees to conditions that never change. The result of that much uninterrupted time, in that much isolation, is something we have not yet found words for. Science is still building the tools to see it clearly. Now ease your attention outward from the buried interior. Let it drift toward the edges, where the land meets the sea in a way that most maps do not adequately show. Antarctica does not end at a coastline the way most land masses do. Its glaciers do not stop at the shore. They continue outward, sliding off the continental rock, extending over the surface of the Southern Ocean, forming vast floating platforms of ice called ice shelves. These shelves are not thin layers. They are hundreds of meters thick in places, thicker than the tallest skyscrapers in the world's largest cities. And they fringe nearly 3/4 of Antarctica's entire coastline. The Ross Ice Shelf alone covers an area roughly the size of France. The Filchner-Ronne Ice Shelf is larger still. Together, Antarctica's ice shelves cover an area that exceeds many countries. And beneath every square kilometer of them lies a sealed, lightless ocean. An ocean that has received no direct sunlight in some areas for millions of years. This hidden ocean is not a small or marginal feature. It is one of the largest sealed marine environments on the planet. Water circulates within it in slow, looping currents, interacting with the underside of the ice above and the seafloor below. Temperatures hover near the freezing point of seawater, just cold enough to keep organisms from metabolizing quickly, just warm enough to keep them alive. Storms can rage on the surface above. Seasons can turn. Years can pass. And in the dark water beneath the ice, very little changes. The stability is remarkable. The silence is absolute. For most of human history, no one looked into this space. There was no practical way to reach it, and few reasons to suspect it held anything worth finding. Ice shelves are thick. Drilling through them is expensive and technically demanding. It requires months of preparation. Each expedition represents years of planning and significant risk. And when the first cameras were finally lowered through drill holes in 1977, no one was sure what they would find. They found life. Not immediately dramatic life. Not the kind that announces itself in color or motion, but life unmistakably. Organisms anchored to the seafloor, moving slowly through the water column, living out long, unhurried existences in conditions that previous scientific consensus had called nearly uninhabitable. Subsequent expeditions found similar communities elsewhere. Each time, the discovery required a quiet revision of what we knew. The organisms found here are extraordinary in their ordinariness. Sponges with no brain, no nervous system, and no ability to move. Growing in cold darkness at rates measured in fractions of a millimeter per year. Some Antarctic sponges are believed to live for hundreds of years. A few may live for thousands. Their tissues accumulate chemical signatures across centuries, making them living archives of an ocean no human eye ever saw. Starfish move across the seafloor at a pace that makes the word movement feel generous. Their progress can take hours for a distance measurable in centimeters. Fish navigate in total darkness, using sensory systems that detect changes in pressure and water movement, rather than relying on vision. Some carry antifreeze proteins in their blood, proteins that prevent ice crystals from forming in their tissues. Their eyes are often small or vestigial, energy-expensive organs that darkness has made redundant, slowly simplified across evolutionary time. In one expedition, scientists drilled through nearly 900 meters of ice to reach the ocean beneath the Ross Ice Shelf. They lowered a camera into the black water below. The expectation was modest. Sediment perhaps, and the possibility of a few microbial mats. What the camera captured instead was fish. Calm, unhurried fish, swimming in total darkness, in water cut off from all surface productivity, living in an ecosystem that had no obvious right to exist as robustly as it did. The discovery did not make headlines in proportion to what it meant. But among researchers who study these environments, it settled something. Complex life does not require light. It requires opportunity. And beneath the ice shelves of Antarctica, opportunity, while narrow, has persisted for an extraordinary length of time. If the quiet strangeness of this hidden ocean is drawing you deeper into calm, a like on this video helps share these hidden worlds with others. Others who might need exactly this kind of exploration tonight. There is a framework most of us carry without ever examining it. It sits beneath our understanding of the living world, the way a foundation sits beneath a building, so fundamental that we forget it is there. The framework goes something like this. Sunlight arrives. Plants capture it. Animals eat the plants or eat each other. Seasons turn. Cycles repeat. All of life on Earth is organized around this steady input of energy. From a star 93 million miles away. Even in the deepest ocean, even in the darkest cave, this framework shapes what we expect to find. Remove light and life must thin out. Remove it completely, and life must eventually stop. Antarctica's hidden environments exist entirely outside this framework. In the subglacial lakes, in the sealed ocean beneath the ice shelves, sunlight does not merely dim. It is not present at all, not in trace amounts, not filtered through layers of water, not bounced and scattered into a faint ambient glow. It simply does not exist. There are no photons reaching those environments. There never have been. The organisms that live there have no relationship with the sun whatsoever. They did not lose the sun and adapt. They evolved entirely in its absence. The sun, for them, is not even a concept. This forces a fundamental reconsideration of what life requires. The honest answer, delivered quietly by decades of Antarctic research, is that life requires very little. Beyond liquid water, a source of chemical energy and time. That is not a small thing. Liquid water and chemical energy are not trivially available everywhere. But the list is shorter and more flexible than the sunlight-centric model suggested. Photosynthesis is not a requirement. It is a strategy, an exceptionally successful strategy that dominates Earth's surface. But one of several possible strategies, not the only one available. In the absence of sunlight, chemosynthesis provides the foundation. Microorganisms extract energy from inorganic chemical reactions, converting iron compounds, sulfur, hydrogen, and methane into biological matter. They do this slowly. Chemical energy yields less usable power than sunlight. The entire system runs at a reduced pace. Growth is measured in years, not days or weeks. Reproduction is infrequent. Metabolic rates are calibrated to conserve energy across spans of several human lifetimes. In the absence of seasons, something equally profound changes. Seasons are not merely a convenience for organisms that live by them. They are a structuring force. They tell life when to prepare, when to act, when to slow down. Migration, flowering, reproduction, hibernation, all calibrated to seasonal cues, primarily to changes in day length driven by Earth's tilt. Remove the seasons and these cues disappear entirely. There is no signal to grow quickly in summer, no warning to prepare for winter scarcity, no rhythm of feast and famine that drives population cycles. There is only constancy. Cold, dark, pressurized, chemical, and endlessly, almost incomprehensibly stable. What constancy does to biology is subtle. It takes a long time to become apparent. Without seasonal cues, circadian rhythms lose their usefulness. Those are the internal biological clocks that regulate daily cycles. The timing of hormone release, the opening of flowers. Maintaining those clocks costs energy. Energy spent on a clock that has no time to keep is energy wasted. Across evolutionary time, organisms in seasonless environments simplify these systems, redirecting the resources toward maintenance, reproduction, or basic metabolic function. Vision follows a similar logic. Eyes are expensive organs. They require significant neural infrastructure to interpret their input. In total darkness, they provide no information at all. The evolutionary pressure to maintain functional eyes in permanently dark environments is, over millions of years, essentially zero. What replaces them are the other senses. Chemical detection becomes the primary means of navigating, of locating food, of finding mates, of understanding the environment. the ability to sense gradients in dissolved compounds at extraordinarily fine resolution. Pressure sensitivity provides spatial awareness in the absence of sight. The ability to detect changes in water movement and vibration. These systems are not compensations for lost senses. In the organisms that rely on them, they are primary, dominant, highly evolved, the main way the world is experienced. The result of all of this, accumulated across millions of years, is biology that operates by different rules, not alien rules. The chemistry of life is the same. The genetic code is the same. The fundamental processes of metabolism and reproduction follow the same logic. But the timing, the scale, the priorities, and the sensory experience of being alive are so different from anything familiar that they challenge our intuitions. And that challenge is, if you let yourself sit with it, one of the most genuinely extraordinary things that Antarctic science has to offer. There is a phrase that appears again and again in scientific papers about Antarctic biology. It appears in introductions, in discussion sections, sometimes in conclusions, almost as an afterthought, as if the researchers feel slightly embarrassed by how often they must repeat it. The phrase is this: conditions previously thought to be uninhabitable. It has appeared so many times in Antarctic science literature that it has stopped feeling surprising. It has started feeling like a structural feature of the field, a phrase that signals not a surprising exception, but a pattern so consistent it has become expected. Antarctica has been quietly dismantling our assumptions about where life can exist for the better part of a century, not through a single spectacular revelation, not through the discovery of a large unknown animal that rewrites textbooks, but through the accumulation of evidence from places where the expectation was absence, from environments where the question was not what lives here, but whether anything could live here at all. And again and again and again, the answer has been yes. The dry valleys of Antarctica offer one of the earliest and most striking examples. These are regions in the Transantarctic Mountains, where the topography prevents the ice sheet from advancing, leaving behind landscapes that receive almost no snowfall, scoured by wind until they resemble the surface of Mars. Temperatures stay below freezing for most of the year. Liquid water is nearly absent. Ultraviolet radiation, reflected from ice and clouds, is intense. There is no vegetation, no soil in any meaningful sense, no insects, no audible sound of any biological origin. By every intuitive measure, these valleys should be sterile. They are not. Within the rocks themselves, not on their surfaces, but inside them, in microscopic pores and cracks, where small amounts of light filter through translucent minerals, communities of microorganisms exist and function. These endolithic microbes live protected by the rock, protected from the worst of the cold and radiation. They photosynthesize using the tiny quantity of light that penetrates their shelter. They may divide only once every several years. During harsh periods, they may go dormant for decades, suspending metabolism to near zero, waiting for conditions to shift before resuming. What they are doing, observed on the timescale of a human life, looks almost exactly like nothing on the timescale of the rock they live inside. It looks like thriving. In the waters surrounding Antarctica, evolutionary adaptations reach remarkable levels. The ice fishes, a family found only in Antarctic waters, have no hemoglobin. Hemoglobin is the molecule that carries oxygen through the bloodstream. In virtually all vertebrates on Earth. In ice fishes, it is simply absent. Their blood is clear and colorless. At first encounter, this seems like a profound disability. Hemoglobin exists because oxygen transport is essential to aerobic life. Remove it, and how does the animal breathe? The answer lies in the oxygen-rich cold waters of the Southern Ocean. Elevated oxygen content combined with enlarged hearts, expanded blood volumes, circulatory systems scaled to move large quantities of oxygenated blood efficiently. Together, these compensate for the absence of hemoglobin. With remarkable effectiveness, the ice fishes are not struggling. They are not barely surviving. They are thriving, as they have been for millions of years. Since Antarctic waters cooled enough to make their unusual biology advantageous. Their antifreeze proteins bind to small ice crystals in the blood and tissues, preventing those crystals from growing large enough to cause damage. These proteins evolved independently in Antarctic fish and in Arctic fish, on opposite ends of the Earth. A case of convergent evolution so precise that it provides some of the clearest evidence available. That evolution, given similar pressures, tends towards similar solutions. Beneath the ice shelves, the pace of discovery has been slower, simply because access is so difficult. But each expedition has returned with evidence that challenges prior assumptions. In one expedition, beneath the Filchneron ice shelf, a camera lowered through hundreds of meters of ice captured something unexpected. A sponge, a multicellular animal, not a microbe, attached to a boulder more than 1,000 kilometers from the nearest open water. There was no clear explanation for how an animal of that complexity could survive there. The discovery prompted significant debate and further investigation. Hypotheses were put forward about long-range particle transport, about local chemosynthetic production not yet detected, about energy economies more efficient than previously modeled. What was not seriously entertained was the suggestion that the sponge was not there. It was there. It was alive. It had been there in all likelihood for a very long time. The pattern from all of these discoveries taken together is not dramatic. There is no single moment of revelation, no watershed finding that overturns everything that came before. The pattern is quieter and, in many ways, more interesting. It is a pattern of consistent surprise, of expectation meeting reality, and finding reality to be more generous than expectation predicted. Every time a new Antarctic environment has been sampled, life has been found. Not always complex life, not always abundant life, but life. Active, metabolizing, reproducing, adapting life. Doing in the cold and the dark and the pressure what life has always done. Finding a way to continue. What part of this hidden world moves you most? Leave a comment below and let us know. The conversation in these quiet spaces is one of our favorite parts of Info Jungle. When scientists speak about evolution in isolated environments, they are describing a process most of us never directly observe. The timescales involved exceed not just individual human lives, but the entire recorded history of human civilization. By orders of magnitude, evolution on the surface of Earth is typically driven by disruption, by the arrival of new competitors, the disappearance of old food sources, the cycling of ice ages, the shifting of sea levels, the random catastrophes that have punctuated life's history since the beginning. Under these conditions, adaptability is often more valuable than specialization. The ability to respond quickly to unexpected change tends to be favored. Because the environment shifts frequently, beneath Antarctica's ice, the opposite is true. The defining feature of these environments is not disruption, but continuity. Temperature does not swing. Seasons do not exist. New species do not arrive. Food sources do not fluctuate wildly. The same chemical gradients that sustained life a million years ago sustain it today, in roughly the same proportions, with roughly the same availability. In this context, the evolutionary logic inverts. Flexibility carries a cost. Maintaining the biochemical machinery of adaptability requires resources. Specialization in an environment that will not change is more efficient. Organisms that precisely fit their niche persist better over the long term. The result of this pressure, applied across millions of years, is organisms that are extraordinary in their specialization. Their enzymes function optimally at temperatures that would destroy most surface proteins. Their cell membranes maintain fluidity at cold that would render others rigid. Their metabolic pathways extract the maximum possible energy from minimal chemical input. Efficiencies that, in some cases, approach theoretical limits for biological systems. These are not organisms that are barely getting by. They are organisms perfectly suited to conditions we find incomprehensible. The conditions suit them. We are the ones struggling to imagine it. Genetic drift plays a particularly important role in these isolated ecosystems. In small populations, random changes in gene frequencies can have outsized effects. When a population numbers in the thousands rather than the millions, random mutations that become common simply by chance can spread, without necessarily being driven by natural selection. Over thousands of generations, these accumulated changes produce real divergence. Organisms that differ significantly from their ancestors, even in the absence of strong directional selection. Combined with genuine selective pressure toward cold tolerance and metabolic efficiency, genetic drift produces lineages that diverge quietly from the broader tree of life. Genetic sequences recovered from Antarctic subglacial environments confirm this. Some sequences match known groups of bacteria closely enough to suggest common ancestry. Others diverge so early from any known lineage that they imply millions of years of independence. Branches on the tree of life that separated from familiar forms. Before the first humans existed, before the first primates appeared, before the dinosaurs disappeared, and have been quietly following their own trajectory ever since. These sequences do not come from dead organisms preserved in ice. They appear alongside RNA. RNA degrades rapidly in the absence of active cellular metabolism. Its presence indicates living organisms actively expressing genes in real time. Whatever these deeply divergent lineages are, they are not relics. They are alive. They are doing something right now, in the dark beneath the ice, something they have been refining for longer than most other life forms have existed. There is also a concept worth sitting with here. The idea that stability can be a form of evolutionary protection. On Earth's surface, extinction events have repeatedly swept away enormous proportions of life. The asteroid that ended the Cretaceous period eliminated perhaps 3/4 of all species. Previous extinction events were larger still, but these catastrophes were surface events, events shaped by the sky, by collisions, by volcanic eruptions, by climate changes from the atmosphere downward. Beneath 4 kilometers of Antarctic ice, none of these events registered with the same force. The stability that seems like a limitation in evolutionary terms. That absence of disruption that prevents rapid adaptation may also be the reason some lineages beneath the ice have survived uninterrupted through extinction events that reshaped almost everything else. In environments where direct observation is rare and fleeting, where every expedition represents years of planning and only weeks of data collection, science must learn to read the world indirectly. This is not unusual in the history of science. Some of the most important discoveries have come not from direct observation, but from noticing that something in the data does not behave as expected. Antarctica's hidden ecosystems are understood largely through this kind of indirect evidence, through chemistry that implies processes, through genetics that implies organisms, through physical signals that imply activity, and through patterns that persist across multiple independent data sets, in ways that resist explanation without invoking life. Water chemistry is among the most reliable indirect indicators available. When samples are drawn from subglacial lakes or beneath ice shelves, researchers analyze dissolved gases, isotopic ratios, nutrient concentrations, and the chemical byproducts of metabolic processes. These measurements can reveal biological activity, even when no organisms are visible, even when no genetic material has been successfully recovered. Methane, for example, can be produced by geological processes, by the slow breakdown of organic material in sediments. But biologically produced methane carries a distinctive isotopic signature, one that differs from geologically produced methane in ways that careful mass spectrometry can detect. In several Antarctic subglacial samples, methane isotopes lean toward the biological. This does not definitively prove microbial activity, but it strongly suggests it. And it is not the only chemical signal pointing in the same direction. Nitrogen cycling leaves traces in water chemistry that are difficult to explain without biology. Microbes that process nitrate, nitrite, and ammonia do so in predictable ways. When those patterns appear in subglacial water samples, they imply the presence of active microbial communities. The imbalances seen in some Antarctic samples are not consistent with geological chemistry alone. Something biological is altering the nitrogen budget. Multiple independent research teams have observed this in multiple locations. The signal is real. Environmental DNA sequencing has transformed the ability to detect life without needing to capture and study whole organisms. Every living cell sheds genetic material into its surroundings. Fragments of DNA and RNA released through normal cellular processes, through death and decay, through reproduction. Water samples from beneath the ice contain these fragments. Sequencing them reveals the genetic diversity of the community that produced them. The picture that emerges from Antarctic subglacial samples is richer than any model predicted. Hundreds of distinct genetic lineages appear in single water samples. Many are recognizable as known types of microorganism. Some are not recognized at all. They do not match any sequence in any global genetic database. They recur across different samples from different locations at different times, ruling out contamination as an explanation. They are, by any reasonable interpretation, real organisms. Organisms that have not yet been formally described. Physical signals add another layer of evidence. Sensors designed to measure water circulation have detected subtle disturbances, localized disturbances in flow patterns that do not correspond to currents driven by melting and freezing at the ice-water interface. These disturbances are small, not the dramatic signature of a large animal passing through, but gentle fluctuations in particle distribution and water movement that repeat in ways inconsistent with purely physical explanations. Researchers are careful with these signals. They are not interpreted as definitive evidence. They are flagged, noted, and used to motivate further investigation. But they do not go away when re-examined. They persist. Acoustic data from instruments beneath the ice occasionally return unexpected signals. Faint, irregular acoustic patterns that do not fit known categories of ice movement or geological activity. These are not the calls of unknown animals. They are simply patterns that require more data before interpretation. Researchers note them and set them aside in a category of things that are not dismissed. They are remembered. There are also statistical shadows, patterns that emerge from comparing what models predict against what sampling finds. When a model predicts certain concentrations of dissolved compound in a subglacial lake, based purely on known geological inputs and chemical reaction rates, and measured concentrations consistently deviate in the same direction, something is influencing the system that the model has not accounted for. The most parsimonious explanation, in environments where biological activity is confirmed, is that organisms are consuming or producing those compounds, not dramatically, not in quantities that would strike anyone as impressive, but consistently, measurably, in ways that require explanation. These statistical shadows accumulate over years of data collection into something that functions as indirect but genuinely compelling evidence. What the totality of these signals suggests, taken together with appropriate caution, is that Antarctica's hidden environments are biologically active, chemically dynamic, and more diverse than the simplest models predict. The picture of life beneath the ice that current science has assembled is a sketch, not a portrait, broadly accurate in its outlines, almost certainly incomplete in its details. The sketch is not wrong. It is simply not finished. And the blank spaces within it are not empty. They are full of things that patient, careful, unhurried science will eventually see. For more quiet journeys into the world's most hidden and extraordinary places, hit the notification bell and let Info Jungle guide you. There is always something waiting to be gently uncovered. Naming a living thing is not a casual act. It is the culmination of a process, one that requires observation, repeated sampling, careful comparison, analysis of structure and genetics, and behavior and ecology, and finally, the construction of a formal description, detailed enough to allow other scientists anywhere in the world to recognize the same organism. A species is not defined by a single photograph or a fragment of genetic code. It is defined by a body of evidence assembled with care. In environments where direct observation is rare, where sampling is constrained by technology and ethics and sheer difficulty. This process slows dramatically, and in Antarctica's hidden worlds, it has slowed to a pace that matches the ice itself. The result is a category of life that is genuinely unusual in modern science. Organisms that are known to exist, whose existence is supported by multiple independent lines of evidence, but which have not yet been formally described. Not hypothetical organisms, not speculation, but real biological entities, whose genetic material has been sequenced, whose chemical signatures have been detected, whose effects on their environments have been measured, and which nonetheless remain unnamed, because the full description required to name them has not yet been possible to complete. Some of these unnamed organisms are microbes, bacteria and archaea, whose genetic sequences place them in entirely new branches. Branches of the microbial tree of life that diverged before Antarctica was sealed beneath ice. Branches that have been evolving independently for tens of millions of years. Their metabolic capabilities can be inferred from their genes. Some appear to use energy sources that are rare or unknown in studied environments. Some encode enzymes that do not match characterized forms, suggesting biochemistry that has diverged significantly from what surface organisms use. Without intact cells to study, without cultures to grow and observe, these organisms remain understood only as possibilities. Real possibilities grounded in data, but possibilities nonetheless. Beyond microbes, there is the possibility of small, soft-bodied multicellular organisms. Animals without shells, bones, or mineralized structures of any kind. These are difficult to detect, even in well-studied marine environments. They leave almost no fossil record. Soft tissue degrades quickly, without exceptional conditions for preservation. They are easy to overlook in water samples, easy to miss in video footage, easy to undercount in studies not specifically designed to look for them. In the deep sea, entire phyla of small soft-bodied animals went largely unstudied until the second half of the 20th century, not because they were rare, but because finding them required techniques that earlier researchers had not applied. In Antarctica's sub-ice marine environments, meiofaunal communities have barely been examined. Meiofauna are organisms that live between grains of sediment on the seafloor. The sediments beneath the ice shelves may harbor communities of tiny animals, worms, crustaceans, and organisms with no familiar common names, living there for millions of years, slowly processing organic matter, completing nutrient cycles, occupying niches that no surface ecologist has yet described in these environments. Their populations would be sparse by surface standards, their movements slow enough to appear absent during a brief camera survey, their bodies small enough to pass through collection nets designed for larger organisms. They are hiding in the absence of sight, in the dark, beneath kilometers of ice, exactly where you would go if you wanted to remain undiscovered. There is also the possibility of organism adapted to exploit the faintest energy gradients, gradients that approach the theoretical minimum for sustaining active metabolism. On the surface, even in energy-poor environments, differences between zones are relatively large. Beneath Antarctica's ice, the available gradients are fainter. The difference between the richest and poorest zones in a subglacial lake may be smaller than anything measured in any studied surface environment. organisms capable of exploiting gradients. This feint would operate at metabolic rates so low, they might register as effectively inert by most detection methods. Their activity would show up not as movement or growth, but as extremely slow, extremely subtle shifts in local chemistry. Exactly the kind of signal that requires highly sensitive instruments and long observation periods to reliably detect. The unnamed creatures of Antarctica's ice-covered world are not waiting to be dramatic. They are not preparing a revelation. They are simply continuing, as they have always continued, in the cold and the dark, at their own pace, on their own terms. The naming will happen eventually, as access improves and instruments become more sensitive. When it does, the names given will belong to organisms that have existed far longer than the scientists who named them. That asymmetry, the vast age of the discovered against the brief life of the discoverer, is one of Antarctica's quietest and most profound gifts to human understanding. For most of the history of the search for life beyond Earth, the guiding assumption was shaped by the only example of life science had available: life on the surface of Earth, powered by sunlight, dependent on moderate temperatures, requiring liquid water at the surface, organized around chemistry that feels recognizable to us. Planets were evaluated for habitability by how closely they resembled Earth. Venus too hot, Mars too cold and too dry. The outer solar system too far from the sun for surface liquid water. The list of places where life could exist was short. Antarctica has been systematically expanding that list for decades, not because it has provided evidence of life on other worlds. It has not, but because it has demonstrated on Earth that life is possible in conditions the old model would have excluded. Life beneath kilometers of ice, life without sunlight, life without seasons, life drawing energy from chemistry rather than from a star, life persisting for geological timescales in conditions that challenge human technology. Each of these demonstrations widens the range of environments that could host life. And as that range widens on Earth, it widens in our conception of what is possible elsewhere. Europa is a moon of Jupiter, approximately the size of Earth's own moon. It is believed to have a global ocean of liquid water beneath an icy shell, a shell that may be tens of kilometers thick. The evidence for this ocean comes from multiple sources. Magnetic field measurements detected signatures consistent with a conducting fluid beneath the ice. Geological activity on the ice surface suggests tidal flexing of a liquid interior. And observations by the Hubble Space Telescope and later the James Webb Space Telescope captured plumes of water vapor erupting from the surface, material from the subsurface ocean being ejected into space. Europa's ocean is not a speculation. It is a near certainty. Enceladus is a small moon of Saturn. The Cassini spacecraft flew through active geysers erupting from its south pole and sampled their contents directly. The geysers contain water vapor, ice particles, organic compounds, molecular hydrogen, and silica grains. Silica grains best explained by high-temperature reactions between liquid water and rock. These are collectively the same ingredients found near hydrothermal vents on Earth, the same type of environment where chemosynthetic ecosystems flourish. Enceladus currently has the chemical conditions that on Earth produce active ecosystems. The question is whether they produce active ecosystems on Enceladus as well. Antarctica provides the closest natural analog available for studying these environments. Not a perfect analog. The temperatures are different. The chemistry differs in important ways. The geological context is not the same, but the fundamental parameters overlap significantly. Liquid water sealed beneath ice. Darkness, pressure, chemical energy from rock-water interactions. Isolation from the surface over long time scales. If science can understand how life functions in Antarctica's subglacial environments, that understanding will directly inform how we search for life on Europa and Enceladus. The lesson Antarctica offers to astrobiology is not the lesson of a hospitable place. It is the lesson of a demanding place that life inhabits anyway. That lesson is ultimately about the flexibility of life, its willingness to occupy conditions we would not choose, to persist in environments our technology cannot easily reach, to continue through timescales our intuitions cannot hold, using energy sources our biology does not employ. If life on Earth is this flexible, there is no principled reason to assume that life in the universe is confined to the narrow band of conditions that feel familiar to us. Antarctica's ice does not make the universe teem with life, but it removes the confidence with which we could have said it does not. If life beyond Earth resembles Antarctic subglacial life, it will not announce itself in ways current detection strategies are designed to find. It will not produce oxygen in quantities that spectroscopes can detect from space. It will not create visible surface structures. It will be quiet, chemical, indirect, detectable only through sensitive measurements of water chemistry and genetic material. The instruments that could find such life have not yet been sent to the moons where it might exist. But Antarctica is teaching scientists what those instruments need to measure, what signals they need to be sensitive to, and what patience the search will require. The lessons are being written now, in cold water samples and genetic sequences and chemical measurements, by researchers working in some of the least hospitable field conditions on Earth, contributing to a search that extends far beyond any single planet. It would be reasonable to ask, given how long Antarctica has been known to science, why so much of its biological life remains undescribed. Satellites have mapped its surface in extraordinary detail. Ice cores have been drilled and analyzed. Research stations have been continuously operated on the continent for decades. And yet the biological systems that exist beneath the ice remain largely unknown. The question of why is not a simple one. The answer involves layers of challenge that reinforce each other, compounding the difficulty far beyond what any single obstacle would produce alone. The most fundamental barrier is physical. Antarctica is not merely remote in the sense of being far away. Though it is that, it is remote in a deeper sense. Even once you are there, the environments that matter most for biology are not accessible. They are beneath the surface, beneath hundreds or thousands of meters of ice, in water bodies with no connection to the atmosphere, in marine environments roofed by ice shelves hundreds of meters thick. Every sample requires drilling through ice first. Every camera must be lowered through a hole no wider than the drill that made it. Every measurement requires instruments that can survive cold and pressure and darkness, conditions that break most equipment not specifically designed for the purpose. The drilling itself is a significant undertaking. A single borehole through 4 kilometers of ice requires specialized machinery. Machinery that must be transported to field sites far from any permanent infrastructure. Assembled, operated, and maintained in temperatures that freeze hydraulic fluid, that shatter metal. The drilling fluid used to keep the hole from collapsing must be carefully selected to avoid contaminating the environment below. The entire operation must follow protocols strict enough to ensure that whatever is found in samples came from the subglacial environment, not from something introduced from the surface. All of this takes years to plan, months to execute, and produces, at the end of a successful drilling campaign, a hole perhaps 30 centimeters in diameter, through which science must try to understand an environment the size of a large country. Technology presents additional constraints even once access has been achieved. Remotely operated vehicles must fit through the borehole, must carry their own power, must navigate without GPS signals, blocked by kilometers of rock and ice above. Must do all of this for hours or days at a time before being retrieved. Their range is limited. Their observation footprint is small. The area of seafloor beneath Antarctica's ice shelves they have collectively surveyed represents a fraction of what is present. Most of the seafloor beneath the ice has never been seen by any camera. At any time, field season constraints impose their own limits. The Antarctic summer lasts roughly 4 months. Setting up a drilling operation, reaching the subglacial environment, collecting samples, and recovering equipment must all be accomplished in this window. If a storm grounds the aircraft supplying remote field camps, weeks can be lost. If equipment fails, the season may end before it can be replaced. Researchers often spend more time preparing for a single field season than the season lasts. And after returning with samples, analysis may take years, so that a drilling campaign from one decade may yield its most significant insights in the next. There is also the matter of priority. For much of the history of Antarctic science, biological questions competed for resources. Glaciology, atmospheric science, oceanography, and climate research had compelling global relevance. They were supported accordingly. The infrastructure built to serve these disciplines was not always adaptable for biological exploration. The transition from ice as an archive of atmospheric history to ice as a barrier to biological environments required new science, new engineering, new protocols, new institutional frameworks, and new funding streams. A transition that has taken decades and is still ongoing. Perhaps most importantly, the life beneath Antarctica's ice does not make itself easy to find, even when conditions for finding it are met. It is sparse, slow-moving, chemically subtle, and often microscopic. It does not produce the visual signals that make biological exploration rewarding. There are no shoals of fish visible through the water column, no forests of kelp anchoring an obvious ecosystem, no charismatic megafauna drawing the eye to productive habitats. There are microbes in water samples that require molecular analysis to detect, chemical gradients that require sophisticated instruments to measure, genetic sequences that require bioinformatic pipelines to interpret. The science of Antarctic biology is the science of finding extremely quiet signals in extremely noisy data from extremely difficult environments. That it has found as much as it has is a testament to the ingenuity and patience of the researchers. That so much remains unknown is simply an accurate reflection of how difficult the problem is. We have traveled a long way tonight, through the wind and the dry cold of the surface, down through 4 kilometers of compressed time and preserved atmosphere, into sealed lakes that have not touched open air since before our species existed, through the lightless ocean beneath vast floating shelves of ice, into the chemistry and genetics and evolutionary logic of organisms that have made their lives in conditions we can barely visit. It has been a slow journey, as Antarctica demands. There was no rushing it. The continent does not allow rushing. What we have found is not what most people imagine when they think about hidden life. It is not dramatic. It is not the kind of discovery that fills headlines with large claims. It is something quieter, and in its quietness, more genuinely astonishing. It is the picture of life as a property of the universe, rather than a property of comfortable conditions. Life as something that does not require our familiar inputs, does not need our seasons or our sunlight or our moderate temperatures, does not ask for the abundance that surface ecosystems take for granted. Life as persistence, life as efficiency, life as the inexorable tendency of chemistry to organize itself into processes that continue. Beneath Antarctica's ice, right now as these words reach you, organisms are metabolizing in complete darkness. Chemical reactions are proceeding in sealed lakes, lakes that have not been touched by surface processes for millions of years. Genetic lineages that diverged from the rest of life on Earth before the Himalayas rose to their current height are completing their generation cycles at a pace so slow that a human lifetime would not capture a single complete round. And in vast stretches of seafloor beneath ice shelves that no camera has ever surveyed, organisms that have not been named, not been described, not been counted, are living out their quiet, extraordinary existences, exactly as they have been doing for longer than our species has been capable of wondering about them. They do not know we are curious about them. They do not need our attention to continue. They are not waiting to be discovered. They are simply there, as they have always been. Patient, persistent, perfectly suited to a world we can barely reach. And yet there is something worth noticing in the gap between our urgency and their patience. We arrive at Antarctica with plans and timelines, with funding windows and field seasons, with the pressure to return data that justifies the cost and risk of being there. The continent receives us with complete indifference. The ice does not accelerate on our behalf. The organisms in the subglacial lakes do not surface to meet us. The unnamed creatures do not arrange themselves for the camera. Everything we have learned about life in these environments has been extracted from unwilling darkness by instruments and techniques invented specifically for the purpose, by researchers who have spent decades developing the patience to accept that answers come slowly here, if they come at all within a single career. That gap between the pace at which we want to understand and the pace at which understanding becomes available is itself one of Antarctica's most valuable teachings. Most of modern science operates at a speed calibrated to human attention spans. Results are expected within years. Publications are measured in months. Antarctica refuses this rhythm absolutely. It operates on the timescale of the organisms within it. It requires that science slow down to match the subject it is studying. And when science does slow down, when researchers commit to incremental questions and long observation windows, what they find has a quality of depth that faster work rarely achieves. There is also a dimension to this story that is easy to overlook: the human dimension. The researchers who study Antarctica are not ordinary field scientists. They are people who have chosen to work in one of the most demanding environments on Earth, often for months at a time, separated from family and ordinary life, in conditions where mistakes have serious consequences, where the scientific rewards are measured in fragments: a water sample, a genetic sequence, a chemical reading that may or may not mean what they hope it means. The scientists who have dedicated themselves to understanding life beneath Antarctica's ice have done so knowing that the answers they seek may not be available within their lifetimes. They are contributing to a multi-generational project of understanding. There is something admirable in that, something that resonates with the patience of the organisms they study. Antarctica rewards curiosity, slowly and on its own terms. It gives up its secrets in fragments, in chemical traces and genetic shadows and indirect signals. It does not offer revelations. It offers refinements, each one small, each one a slight expansion of the map of what life can do and where life can be. Consider the full arc of what we have traced tonight. We began at the surface, where the wind and the cold establish a context for everything that follows. We descended through the ice, past layers of preserved atmosphere and compressed geological time, into sealed lakes untouched by the surface world for longer than our species has existed. We drifted outward to the margins, where the ice extends over the ocean and creates vast, lightless marine environments. We explored the biology of life without seasons, without sunlight, without the circadian rhythms that characterize almost every organism most of us encounter. We followed evolution into complete isolation and watched it produce, across geological time, organisms of quiet strangeness. We read the indirect signals, the chemical traces, the genetic fragments, the statistical anomalies. We named the unnamed, at least in the sense of acknowledging their existence. And we traced the line from Antarctica's ice to the frozen moons of distant planets, where similar conditions may be hosting similar life right now. Undetected, patient, waiting for instruments sensitive enough to find them. That is a long journey, and you have made it. The continent has not changed in the time it took. The ice above the subglacial lakes is exactly as thick as it was when we started. The organisms in the dark water beneath the ice shelves have not moved perceptibly. The unnamed creatures in the sediments are still there, still filtering, still slowly completing their slow lives. Antarctica endures our attention with complete equanimity. It will endure our absence with equal composure. It has no investment in being understood. It simply continues, as it has always continued, at its own pace, on its own extraordinary terms. Somewhere far south of wherever you are right now, the ice continues. It does not care about being explored. It does not care about being understood. It is simply doing what it has always done: preserving, insulating, pressing down with enormous weight on a world that has learned, over immense time, to endure that weight with extraordinary grace. And beneath it, in the cold and the dark, the life that has made its home in those conditions continues too. Without urgency, without seasons, without any need for the kind of attention we are so compelled to pay. That patience is perhaps the deepest thing Antarctica has to teach. Not a lesson about extremophiles or chemosynthesis, or the habitability of icy moons, though all of those are real and important. A lesson about time, about what becomes possible when the pressure to change is removed, and the only requirement is to continue. About what life looks like when it has been given millions of years, to find its precise and perfect fit. About what it means to be so well adapted to your environment that the very concept of struggle disappears, replaced by something more like alignment, a fit so complete that existence requires no effort, only continuation. We are not those organisms. We are the ones who arrive with drills and cameras and questions. We are the ones who feel the urgency, who push against time constraints and the limits of our instruments. We are the ones for whom Antarctica is difficult, demanding, and ultimately humbling. And perhaps that is exactly the right relationship to have with a place like this. Perhaps the continent's value lies not just in what it contains, but in what it does to us when we try to approach it carefully, in the patience it demands, the assumptions it dismantles, the scale it imposes, the quiet it insists upon. There are not many places left on Earth that can do all of that. Antarctica is one of them. It deserves and rewards our most careful attention. Antarctica does not rush. Neither should you. Let whatever quiet this exploration has offered settle where it needs to settle. The questions it opens do not need to be resolved tonight. They are the kind best carried gently, turned over occasionally, and returned to when the pace of things allows. The creatures beneath the ice will keep. They have always kept. An info jungle will be here whenever you are ready for the next slow journey into the parts of the world still waiting to be gently, carefully, curiously understood. If this journey brought you peace, curiosity, or simply a few quiet hours of wonder, we would love to have you stay. Subscribe to Info Jungle. Leave a like so others can find their way here too. Share your thoughts in the comments below. For more explorations like this one, calm, unhurried, and full of the world's quiet mysteries, hit the notification bell. We will see you on the next one.